the HIV group, for an hour, 60 minutes. And then we will offer individual sessions again if you have any one-on-one questions or just need help with something. And the HIV group will continue for the last, I think, 30 minutes of this.
Something like that.
Something like that, exactly.
Well, thanks very much. And again, it's a pleasure to be here. Okay, we've got 60 minutes of terror here. And what I'm actually going to try and do is run through a fair amount of stuff here in the next 60 minutes. We may get all the way through my set of slides, after which we could do individual HIV stuff or not. We'll see how it goes. I'm going to take this in a fairly free-form manner, assuming that most of you guys don't actually have a deep knowledge of or interest in HIV. And so I'm going to be talking a little more generally in some cases. Again, thanks to people who made this possible, and who do a bunch of the work, and of course again to the NIH. I should mention again, we're at National Laboratory whose mission is national defense, but we're all academic scientists on soft money, so funding is always an issue. Keep in mind, our slides are up on the Google Drive for the that Sheridan is managing for the IEDB. We also have them separately here with some data files and a little bit of software, and I'll talk about that. Okay, so basically, we're going to talk about the database. I'm going to show you how we search through it, and a couple of examples of how we make the data available. And I'm going to run through some tools, some of the more general, specific and general purpose tools that we have. And please shout out when and if you have a question, because it lets me know that you're there. As again, this at this time URL we have some software which is important for looking at trees and sequences. You may, if you're a serious bioinformatician, you already have things to do these tools to do these tasks. If you don't, you should. And the slides and some data files are there too. Okay, you've seen this page before. This is the introductory page for the HIV sequence database. Yesterday we spent most of our time here on immunology. Today we're going to spend most of it on the sequence database. And the front page here shows, again, this is all multiple ways in. We've got the drop-down things and the toolbars, which get you to the pages which have the list of essentially the same things that are on the toolbars with a little more explanatory text. The things I'm going to go through briefly are the search interface, geography interface, and some of the sequence tools. Like any other good website, we have facts. We have all kinds of facts, including, my favorite is how to cite this database. You always get these plaintive messages. I'm writing my thesis and I need to show this figure from your database. Can you please give me permission? So, sorry, go here. It's all good. And this presentation will eventually show up here. So if you lose track of all the other URLs, keep track of the hip.lenl.gov and you can find it again. Likewise, questions. There's always somebody here. Sometimes it's me. Sometimes it's Brian. Sometimes it's Jenny. There are various people, but there's always someone happy to answer questions. Okay, so we're going to run you briefly just so you know what we do and what we're here for. I'm going to run you through the search database. What you can get from us are sets of aligned or unaligned sequences. You can get them aligned, and you can get subsets in a variety of ways. And I'll go through just briefly how you get it. Once you get these things, there are things you can do with it. I'll show you some of those. One of the questions we get a lot is, can you please send me all the HIV sequences you have in your database? And we typically say something like, sure, you want all 600,000 in a FASTA file? And they say, no, because typically what people want when they want all the sequences is they want a particular region, they want a particular subset, they have a particular question they want. And it turns out that curation is important and good data are important. So one of the things we like to help people with is how to get a good data set to answer the question that they want. And typically it's not everything in the database, because once you have, be careful what you ask for, because you might get it. But in any case, I'll go through this and I'll go through some of these, and I'll note that there's, again, help at Hogwarts, IEDB, or the HIVDB. There's help available in various places. I'm going to run you through just live a search for all the HIV database sequences from whole genome sequences from Brazil, and show you what to do with that sort of alignment. Again, I'm going to be focused mostly on general purpose sort of sequence stuff. Now, how many people here actually deal with alignments on a regular basis? Sequence alignments? Anybody? Everybody? 1, 2, 3, 4, 5. Okay. There are some things that you need to think about, and some people think about them and some people don't. And I'm going to tell you what I think about it, and why it's important. Key points are that whenever you have sequences, you need context, and you need to, and the phylogenetic tree is a good way of getting context. You need to look at your data, and this sounds really dumb, but a lot of people will download big chunks of data, and they've got their tab delimited format, or their commas, and or their FASTA file, and they don't look at it. You got to look at it. You got to find a way in which you can make some sort of preliminary determinations about it. And we have tools for that too. And I'll go through the geography search interface a little bit too. So the sequence searches, we have this interface. And I'm just going to, you'll have these slides available. I'm going to go ahead and just do some stuff here. So if I can find Firefox. I'll just do it. Are we big enough here? So I can go either here to the search interface, or I can go here to the search and search database. And the groovy thing is we have a bunch of fields, and we have a bunch of expandable fields to search on. So there's really a ton of options. Patient information. The one caveat is that like many other databases, not everything is filled out. So we have fields for everything, but not everybody reports when the infection date was and how far the sample was from infection, or exactly what the patient's CD4 count is, things like that. So that's not always available. But things that are always available are the accession number, because we're getting all these sequences go through GenBank. And I should note that our mode is basically GenBank plus value added. So everything goes into GenBank every month or so. We do a download, pull out all the primate lentiviral sequences, and do a bunch of filtering to exclude synthetic and patent sequences. And then a number of the sequences go into the database with special codes that we've arranged to be in place, and we can parse those out again. But everything goes past a pair of goes through past a pair of eyeballs and through a brain before it's finalized in the database. So, like I said, accession numbers are here. Sequence names, length, sampling year. I'm going to go here and start with a country, Brazil, just because it has a convenient sized data set. and I will note that if you click here on sampling country, that will tell me, you know, well, what is it? Oh, it's the country where it was sampled. That's not much help. But the country codes, if you click through, this is international codes, so you can find out, for example, that SA is not the abbreviation for South Africa. That South Africa is ZA, whereas Zambia, which you might expect, is to be ZA is ZM, but South Africa is where are we? South Africa is ZA for Zuid Africa, as you if you remember your Dutch. In any case, the point being here is you can click through from the fields here, to the help in almost every case. But I'm going to say let's get every complete genome, every HIB one from Brazil, and that'll be enough for now. But if I wanted to get patient information, I can add some stuff to it. I could say let's just say I wanted to get the infection year. I could put a star in here, and that'll get any infection year, but it makes sure that it shows up in the output. And if I want to say the patient sex, let's get that too. So this, in these cases, I'm not saying give me any particular year. I'm not saying give me any particular sex. What I'm saying is show me those results in the output. And so this is kind of groovy. We've got the patient code. We've got the accession number. If you click through on the accession number, it brings you to GenBank, or the GenBank format entry actually, which is helpful to be able to do, because not everything's not always obvious. But for example, you want to go there, fine, you've got that. You can click through to the reference if it's been published. There's PubMed ID. You click right through. We've got sequence names. We've got subtypes. We've got country, which is should be all Brazil. We've got year of isolation. Patient sex, where it's reported, and if it's not always reported, and year it's reported. Click on things. It'll let us sort. We don't have a lot of years here. One key point here is the genomic region. So this is the gene map for HIV, which starts off with the LTR and then goes to GAG and then goes to POL and so forth, all across the genome. So it's positions up there. So we've got basically 9 KB from start to finish. The yellow region is what's sequenced here. And as I said, you can sort by all these, and you can also, and this is really the important thing, if you care about sequences, the reason that you would be here would be you want to get these sequences, download them. And I'm going to rant a lot about sequence identifiers and how it's important to have them robust. Key point being that it's really terrible when you are ready to submit your thesis or submit the paper, and somebody says, "Oh yeah, what was the deal with that one sequence?" And you say, "Which sequence?" "Oh, that one there on the tree." And you say, "Oh yeah, that one. Which one is that?" And if you have to go and chase it back, it can be a time-consuming problem. But if you have an accession number on there, you know where to go. You know where it is. So the default here is we put on the subtype and the country, the year, and the name of the sequence and the accession number. All with the accession number there. There are some other options, but if you download the sequences, they'll be assembled into into an alignment. And I'll talk about that alignment a little bit. Right now we're downloading 402 complete genomes, and if we just click there, we get a FASTA file, which ought to go someplace. Where's it going to go? Yes, look at that, really. So I've opened this in AliView, which is the alignment editor I like to use. There are other ones. This has a lot of advantages, not least it's cross-platform and gives you nice scaling. So, okay, I talk about how great the database is and how we get in line. This alignment is sort of, I don't know, what's a nice word for it? Gappy. It's a little gappy here. And and if we were to zoom in and translate it, we would see that there are a lot of broken codons. Right. So even if this were the right frame, these are not all in the. It's not codon aligned, and it's difficult to translate. And one of the problems with HIV is the sequences are full of indels, and they're not all in the frame. Okay, so most of a lot of the circulating virus is in fact damaged and/or dead. So there a lot of erroneous virus is produced. But you know, everything you see is is no more than one step away from an infectious virus. Right? It was produced the offspring of an infectious virus, but it may be dead itself, which means it may have debilitating frame shifts in it. So it's that's something that has to be dealt with. This alignment is sort of, as I say, gappy and I think suboptimal would be the nice way of putting it, simply because we have, as I said, 600,000 sequences in the database. We don't know which ones you're going to be asking for. We don't maintain all these sequences as an alignment. We maintain them all as pairwise alignments to a reference sequence, and we assemble this alignment on the fly. So this will need some post-processing afterwards. But the point being, it's not, in fact, a terrible alignment. We're at the 3' end of the genome, and there's a bunch of stuff here, and it's a starting point. So going back here, I want to show you one other way in. So the search interface gives you a lot of options, but there's also another way in, and I'm going to show you this partly because it's kind of cool, and partly because it's a little scary. So this is a distribution of subtypes, and the subtypes are, as I think I mentioned yesterday, A through K, but not E, for historical reasons. And how many of you have studied any ecology? Anybody? Any idea of the center of origin theory for species richness? The idea basically is if you're looking for where in particular genus, say, of plants originated, the best guess is it's where you have the most diversity, the highest species richness. Oh. Right? And if you want to know where HIV originated, it's where it has the highest subtype richness, which is in the continent of Africa. And if you want to go down into the continent of Africa, and you want to say, well, where did it in fact originate? It brings you right to the DRC and Cameroon, and there's good actually phylogenetic evidence for that this epidemic started off in chimps in southern Cameroon. That's the source. They found those closely related viruses. But the creepy thing here is you can click through and say, okay, well here's the Congo. This is Congo Brazzaville, I think. And if you go and you can see there's a wide variety of different subtypes and recombinant subtypes. Everything that has more than one letter and a number is a recombinant. But you can also click through here, and it will bring you to the back. There you are at the search interface with these sequences, and you can download them. Now, and we may just go from here, I think. So the other thing you can do besides downloading the sequences is you can make a tree. And I'm going to stress the utility of making trees whenever you're dealing with a sequence set, because really what it, in the best of all possible worlds, you'll have homologous positions of lines, and you'll be making a bona fide phylogenetic hypothesis. But in any case, you're going to be doing a reasonable hierarchical clustering by similarity, which is important to do. So the other thing I'm going to do is I'm going to include subtype reference sequences, and I'll explain why it's important later on. Again, this is the tree interface to pop, I believe, which makes the tree. And there are a couple of model parameter things that we do, but it doesn't make a lot of difference. Key point is I'm including reference sequences, and I'm going to include I'm going to use the subtype J sequence as an attribute. Okay, well that's great. Look at that. It's a beautiful tree. No, but the tree is in fact here. There's a PDF. Give me clustering, or more usefully, in fact, as a Newick tree. And you say more usefully. You say yes, more usefully actually. So this is Newick format, named after the Newick's Lobster Restaurant in southern New Hampshire, where Joe Felsenstein and a number of other people met after the Gordon Conference in like 1986. I've been there. I've been to that restaurant. They make a pretty neat lobster. But the point is, you can go ahead and take this stuff. Let's see. And open it in a program like Figtree, which I recommend highly, and you can then manipulate it. And for example, what I just did was I midpoint rooted it and sorted it from top to bottom. And so what I can see here is that these are I've got some reasonably clustered sequences, and I'm looking at the names here. For instance, if I care about the A subtype, they're all the A's are together there. That's good. All the C's are together there. I've got some B's, and they're there, and they're next to the D's, which is this is all sort of a sensible tree. So even though the alignment is probably imperfect, there's stuff there. It's not too crazy. So that's sort of a general example of the kind of thing you want to do with a query. And I'm going to go back to the slides here. And any questions about what the sort of stuff I've done here and why you might care to do it?
How do you choose your route?
So that's actually a deep question.
I only ask deep questions.
All right. No, so the seriously, it's a genuine deep question, because phylogenetic trees, with certain exceptions, are unrooted trees. Okay, so they don't have a default root. What you have is a relationship of distances, or inferred branch lengths, what have you, inferred between nodes. So you have to know something about the biology of your organism, whatever it is, in order to read it correctly. And the typical way is you have your sequences of interest, and a close relative, or a set, or better, a small set of close relatives. And then you root them on, you root those two subtrees on one another. The only actually reliable and unambiguous way of doing it is to have paralogous trees. If you have, two gene families that are derived from one another by an ancient duplication, you can root them on one another. And the people who root the tree of life use that approach. In this case, I was very simple. I just midpoint rooted it. So the midpoint of the tree. Essentially, add up all the branch. Approximately what you do is you add up all the branch lengths on one side and all the branch lengths on the other, and you find the middle, and read it there. That's and it turns out for HIV, that's not too bad, because the subtypes we have kind of do they approximate a star phylogeny. They all radiated out at about the same time. If you want, you can go ahead and root them on the most closely related chip sequences.
How about rooting on the consensus sequence?
People have done that. It's not that great. And the real problem is it's not robust to long branches. Long branches tend to go towards the root, generally speaking. You can sometimes reading on the consensus is okay, but there's no guarantee. I guess is the bottom line. You really do want to have some sort of biologically relevant time period. So, I should note there's this one sequence per patient thing, which is important because a lot of patients are very deeply sequenced. For one of George Shaw's patients, CH40. They have right now something like 5,000 or 6,000 separate sequences from that one patient. You may not want that for your in a data set. You click this button and you get only one per patient. And there's background info, which is nice to have. That basically throws out everything, all the other fields in the database that are available. So I've already showed you what happens when you make a tree. I'm not going to do that now. So I'll just skip along there. But the point is, you can get a tree out, and you can view it in Figtree, and you can do things with the alignment. One of the nice things I didn't mention is back in the tree tool interface. Is back here. You can download the alignments sorted by the tree, which is helpful. But there's a better way of doing it now, and I will show that to you subsequently. Okay, so I showed you how these sequences are aligned, but they're it's not translatable, or it is, or if you do translate it, it will be give you an unaligned set of amino acids, and every time there's a frame shift, things will go completely south. So special purpose tool here, gene cutter, takes this and turns it into this. So essentially these regions, these variable regions are, for all practical purposes, they're unalignable. Most sequence alignment algorithms assume that positions evolve by substitution mutation, and in these hypervariable regions, that's not how it happens. What you get are episodic insertions of several nucleotides, and with some strong biases, and basically it's not alignable in a ancestral sort of way, in an evolutionarily sensible way. So in this case, we punt and cram them all together in such a way that you have even codons at the ends. And that's not alignable, it's not aligned, but it's compact and you can use it. So I've gone through geography a little bit, and I'm going to skip over these next things. I will note just that for any search, you can generate a histogram of coverage over the genome, and the thing you will almost always find is that there's strong biases on what's actually sequenced. In this case, the parts of the genome that are most of most interest medically are the polymerase gene, which is here because that's where most of the drugs are targeted. So they sequence this region all the time to look for drug resistance, and the envelope gene over here because the variable region is used for essentially counting isolates and also for antibody research. And the other point is it's a database, right? So we're dependent on sampling, and most of the sampling is what's in the database reflects what's been sequenced, and what's sequenced reflects where there are viruses adjacent to sequencing machines. Okay, so although most of the infections in the world are subtype C, most of the sequences are subtype B because subtype B is what's most prevalent in the richer portions of the world. So that's just a general aside, by the way. Think about what your data is and where it comes from. I showed you this, and I just made this point. The thing again to note when you go through in here is there's no HIV in North Africa or the Muslim world. Not a single infection. Well, no, that's not quite true. There's not a single database sequence in the database from North Africa, which is not quite the same thing as saying there's no infection. But there's, I mentioned in some countries there's a strong stigma about reporting, and I believe the official position of the Iranian state is that there is no HIV in Iran. Which is, it's a fine political position, I suppose, but it's not a fine, it's probably at variance with the actual facts. Okay, that's a little riff on geography. I should mention one of the values that we add actually, and it's substantial, is a set of curated alignments, and. When I came into HIV, I was a hot bioinformatician. I knew all there was about sequence alignment, and I could align sequences from here to Sunday. And the more I did in HIV, the harder I realized it was, because of the massive indel problem, because of frame shifts, because of hypermutation. It's a freaking nightmare. And fortunately, we have a lot of people who've actually spent some considerable time dealing with these problems, and we've got these alignments available. And usually, the answer for the person who wants all of our sequences, he doesn't. He wants 5,000 carefully chosen and hand-aligned curated sequences, and that's what we give them. Okay. Just briefly, I'm showing you it's there. You get here from the alignments tool, and you have a choice of various sequence alignments. You can choose what region of the genome you want, and we do have consensus sequences available. And we can every couple 3 years we'll compute a new consensus and say, oh yeah, it's the same as the last one. So we don't change that. We have, as I mentioned, a bunch of tools. You get there by clicking on the tools button, and you can get, if you know where you're going, you can get here. If you don't, you just click there and you get to the tools page, which looks like this. A bunch of tools, and helpfully grouped by straight up immunology, by phylogenetics, variety of different topics and ways to get other things. I'm going to mention just a few, I hope, of these guys. So here's all the tools I would like to talk about today. But those are the ones that I'm going to mention briefly today. And they go from the general ways of thinking, what's going on with a sequence, to convenient ways of just dealing with the data, and so it's a mix of sort of convenience wrapper tools and actual analysis tools. And some of these are for a combination of both. Highlighter, for example, makes a beautiful figure and helps you see what's going on. Okay, first thing here is format conversion. So I don't know. I've been a bioinformatician for 30 years, and I've probably spent 15 years converting files from one format to another, right? And I can't always remember, for example, the difference between PIR format and SLX format and Stockholm. What the heck? What's Stockholm format? Who uses that? Well, it turns out Hummer, which is a pretty an alignment tool that has certain applications, wants Stockholm format. Yeah, I can build a makefile and I can write little Perl scripts to do all the conversion myself, and I do. But you don't always want to have to do that. So this thing takes a wide variety of formats, so you don't have to remember what the heck these are, right? You stick in this, right? You leave that on automatic. You go ahead, you paste your file in here, or you upload it, and you say, I want FASTA file, or FASTA, or I want MACE, or I want, God help us, Nexus interleaved format, and you will get it. Almost all the time. So I mention that simply because this is handy. This is nice. This will can make your life easier and save you hours. So all right, I'm gonna bitch and moan about HIV because I hate it. It's a terrible virus and it's a pain for bioinformatically. So my life is hard, right? Okay. But not as hard as 35 million people who are HIV positive, but nevertheless, I feel a certain sense of grievance. So the virus is a pain. It's got these. It's very small. It's only 10 kb, but it has genes in all 3 reading frames, some of which overlap. Right. So there's this one place here where there are 3 different proteins being translated in 3 different reading frames. There's splicing happening without it. There are a whole bunch of different transcripts. It frame shifts all the time. It's full of errors. It also gets hypermutated as part of the host antiviral response, where the ApoBec3G protein changes G's to A's in certain contexts. And there's a ton of recombination. This is recombinant sequence between 2 different subtypes, and it's very clear that these are subtype A, and this is subtype E, which only exists, by the way, in this recombinant. We don't know where the rest of the virus is. But there are these regions which are ambiguous. They could be either. So turning the breakpoints is incredibly annoying. There are, of course, the usual problems that nobody ever wants to talk about, and since you're mostly informaticians, I urge you strongly to think about the possibility that your laboratory partners have screwed things up. Because it happens. People, stick the pipette into the wrong well. And I know that because I have my own self stuck the pipette tip into the wrong well, or into the wrong tube afterwards. There's cross-contamination. And one of the real problems of bioinformatics, right, is that bioinformaticians believe the experimentalists, right? They just accept this data. It comes in, all this data, the fine, this data, I'm going to analyze the heck out of it. And of course, then the experimentalists get the data back, you get the analysis back, and say, wow, that's great, I don't understand it, but it's very complicated, it must be right. And really, if the data is wrong, then the analysis results are wrong, and you have to consider those possibilities. You always have to ask, this looks funny, could there be a contamination problem? And nobody has ever had a contamination problem in their lab, unless they've looked for it. But you talk to people, it's like, ever had any contamination in your lab? Oh, no, not. You looked? Well, no. Just saying. Okay, the other things that there are problems, generally speaking, in pathogen is people say, oh, I used, I'm talking about this location, residues, 435 to 447. And you say, really, of what virus? Oh, this one. Okay, well, which one is that? I'll talk a little bit about that. And the other thing I mentioned before is accession numbers, sequence identifiers. So, okay, so we have solutions in part for all of these problems. I talked about gene cutter. We also have HIBA line, which does the same thing. I showed you yesterday the pixel tool output, which makes images of the alignment, so you can look at the thing all at once and say, oh yeah, there's a big shift there. And the entropy tool, which I'll show you a little bit later on, which is also extremely helpful in terms of looking for things like, epitopes and mean pressure. We have tools to identify hypermutation, and I mentioned the highlighter, which I'll show you, where you can see where differences per sequence are in sequences. I won't talk about combination identification. I showed you TreeMaker, and I'll touch briefly on the quality control thing. And finally, we have these tools, which are, and I'll talk mostly just about this as an example for bioinformatics, generally speaking. And I'll talk some about this alignment multi-tool, which again is it's a Swiss Army knife for sequence alignments, generally useful not just for HIV. Okay, the point of gene cutter simply is, I mentioned that HIV has 9 different genes, and 2 of them are split by introns, and they're in different reading frames. Actually getting the stupid protein alignment out of it is a pain. We built this tool to solve that problem. And you can throw in sequence data, you'll get back out codon lines, nucleotides, and amino acid translations for all the genes. Only matters to you if you're doing HIV. It doesn't matter if you do HCV because it's one long polyprotein. How beautiful is that? But there are other things like, there are other viruses that are even worse. Hep B, I think, has alternate reading, has reading frames all opposite directions. Some of them overlap. So I'll mention that some of these tools, most of these tools have, paste in, upload, various options, and some of them have a mailback for the long running jobs. If you guys want to try this out for yourself, and I encourage you to do so, these input and output files, which you can compare to one another, are in the Google Drive folder for the IEDB. So what it looks like, so these are unaligned sequences in a pixel format. And it's always nice to look at it and say, oh yeah, these aren't aligned, are they? When you get some sort of stupid result back. And I say stupid, I mean in the nicest possible way. So because I get results back and I say, oh, that's stupid. And what it really means is I was stupid. But so you can take this thing and these sequences, and you can throw them into a standard sequence alignment tool like MAPT. And don't get me wrong, MAPT makes good alignments, generally speaking. But these variable regions, these hypervariable regions, are just hideous, and it doesn't handle them well. You throw it into gene cutter, and you get these all compressed and codon aligned, and it's much more convenient for general use. I showed you TreeMaker, and again, it's a useful thing to use to do to all, make a tree with your sequences, including known reference sequences. This is really important. I can't overemphasize how there are cases where people have taken they have samples from a bunch of different patients, and they've gone ahead, they've made trees for each patient, and they've made interpretations based on looking at those trees for each patient. But what they haven't done is put all their patients together and seen that they are all interleaved with one another, which means that either these patients have been transmitting all from one to another, that it's one big swarm of intermixed infections, which is admittedly possible under some social situations, or they have a problem with laboratory contamination, and somebody has mixed up all their sequences. And if you don't put them all together, you'll never know whether that happened or not. Okay, same. Again, that's how these work. There was a noise. Is there a question? I'm going to. So, if you were to make a tree with a sample input from this tool, and I'm going to mention that because I haven't, I think, is almost all of our tools have a sample input button. So when you click that, it will populate this field with some data of the appropriate input format. And so you can do that, you can put that in there, you can run it, and you can say, oh, this is what the input ought to look like. And, oh, this is the input I would get with my own data. And that just turns out to be really, really helpful. But I'm going to walk you through just a sort of very, very brief sort of tree analysis. So here's a bunch of HIV sequences, and you can see they all start with a letter. That's the subtype, and most of this looks kind of sensible. It's like, oh well, here's an A1, and next to an A2, and that makes sense. Here's this group is Y themselves. There's some other groups, and we've lit up this is what it looks like in Fig Tree if you ask it to highlight the names that start with test, and things that would leap out at me when I look at this tree, even before I colored the branches, is that this branch is kind of long, hanging out there, and that this branch is kind of short, and that this one is down at the base of this otherwise well-defined clade. And it turns out these things each illustrate a particular problem. This branch is long because it's got a lot of changes relative to this one. And if you went and looked at it, you'd say, oh, well, it's full of G to A hypermutation. Okay? So, but it doesn't matter what the cause is. The point is this branch is sticking way out there. There's probably something funny about that sequence. It would look the same if it were misaligned, right? If some section of it were out of sync, the misalignment in your file leads to a bunch of spuriously inferred changes, right? It's a bunch of called differences that aren't different. It's just misaligned. This happens to be the most commonly available. This is one of the first HIV sequence isolated, and it's been cloned everywhere, and it's in labs all over the world. Okay, so that if your sequence that you isolate comes up right next to that, it means either you have a sample from 1983, or there's maybe a possibility that it's a contaminant. You really probably ought to check that out. The other thing that happens here is that this guy's pulled down to the base rather than being nestled within the clade. And in this case, it's because it's a recombinant. That's what recombinants do. So most of this virus is actually C clade, but a big chunk of it is B clade. And the B clade has lots of extra differences that tend to pull it down toward this branch, toward the root of the clade, over toward the home of its other portion. So basically, stuff like this jumps out at you when you make a tree, and it's an important sort of sanity check. Okay, I mentioned this. This is a new tool for us, and it's just sort of handy because there are a lot of things that you can do by yourself, right? And for example, on a side, you can put up your own wallpaper. Okay, it's perfectly possible to wallpaper your own house, and some people do. But it turns out it's hard and difficult, and it's a pain, and you don't always end up with a nice job in the end. Likewise, you can take your alignments and sort them all manually and do various things with them. But sometimes it's nice to have an easy way of doing it, right? You can pay somebody to wallpaper your bathroom. You can use this tool that we built to do a number of different things. The examples I'm going to show you are, so what it does is it sorts alignments by a variety of ways. It lets you rename sequences, again, in a variety of ways without screwing up the format. And you can take slices out of the alignment. You can say, oh, I only want these columns, or I only want these columns, or I have this gene from this set of taxa, and a different gene from the same set of taxa. I'd really like to have them all, to analyze them together, and let you do that too. And there are a bunch of options for sorting. For example, you can sort by tree, you can sort by similarity, you can sort by the starting points of the gene. If you have, for example, short reads, hugely helpful. You can sort by sequence name or part of name. You can decide which sequence you want on the top to be a reference. Just stupid stuff, really handy. And why might you want to sort by tree? Okay, I've talked about that a lot and how that's going to be helpful. Well, so if you take an alignment like this, you might really sort of want to know. Well, it's not clear by looking at this whether this is actually decent alignment or not. Okay, but if you make a tree and you cluster things that are related or look like they're related together, then all of a sudden you can say, oh yeah, this looks pretty good, and that looks pretty good. What's going on over here? Right? Those guys aren't right, and it just makes it obvious so you can fix it. Okay. So that's the sorting and why you might do it, and it can make it'll make a tree on the fly and do it for you if you don't have a huge alignment. And I strongly recommend that you do this if you have reasonable size alignments. Otherwise, you can make a proper tree of your own self and upload it. Yeah, use my tree. So, load a file, choose a model, and what you get is a FASTA file of your sequences in order. And so that's fine. You can download it. You're done. But wait. Now you can say go back with this output. And the groovy thing there is that brings you back to this page, back to the alignment page, with that alignment, that newly processed alignment loaded up, which means you can stack these changes. Okay, so you can say, okay, first I'm going to sort it by tree. Then I'm going to change the names. Then I'm going to take this slice out. So you can keep go back and forth and do a variety of changes. You can, as I say, prune out the sequences you don't want. For example, maybe I decided that that these guys, I don't care about these sequences. Throw them out. Okay, easy to do. Or I'm going to keep just this region. Also easy to do. Or I'm going to keep throw out this region and this region, but keep everything else for all the sequences. All those things are simple enough to do. So that's prune. You can also rename. This is a groovy thing. So you decide you're getting toward the end, and you want to say, oh well, I forgot to put the subtypes in my sequence names. You can do that. You can actually ask it to go back to the database using these accession numbers and put in whatever fields you can extract from there. And finally, you can put things together. So different functions, all convenience. Nothing. There is not rocket science here. It's just hugely helpful. And it's a power tool because you can stack operations. And I'm going to mention again is you can add stuff to the sequence names, and that makes display in the end much easier. And by coincidence, the next tool I'm going to talk about is a tree display tool, where you can use strings and sequence names to color the branches or color and weight the branches. And the point is simply, how do you show what's going on, right? A lot of people aren't going to read your paper. Actually, they're going to look at the pictures. So give them nice pictures. Sequence locator, again, important. I hate to moralize so much, but really, if you're talking about a piece of sequence, if you're talking about an epitope, you have to unambiguously locate it for people, for the readers of your paper, not just now, but 5 years from now. And keep in mind that in 5 years you're going to be a different person too. So you might want to know where exactly that was. So for HIV, we have this beautiful thing where we can put in any bit of sequence, and it will tell us where it is on the genome. It gives us unambiguous numbering, both from within a given gene, right? GP161, it's this amino acids. Also from the start of the genome, and so we have, and this is used by the whole field. And I got to say that HIV is terrible in many respects, but at least the literature is reasonably intelligible because people use this tool and they report things in terms of reference sequence coordinates. And it's not the smallest contribution of Los Alamos. It also gives you alignment, so you say, oh yeah, this is really in fact genuine. All right, I'm going to skip over this variable region characteristics thing. It's kind of cool, but not general purpose. It is kind of cool. So that that's okay. I'm going to almost skip over it. The point being that we pulled a particular region out of sets of sequences, and I've compared the blue sequences to the red sequences. These are all viruses isolated from a single HIV infected subject, a South African woman who rejoices in the euphonious name of Cap256. I'm sure she has a much nicer name, but it's private. So it turns out that when a neutralizing antibody, when she developed a neutralizing antibody, a broadly targeting antibody, it's substantially knocked down these viruses, but these guys were escape forms, and you can tell by looking at the epitope, or the region that this antibody targets. There's substantial differences between these guys. These are mostly earlier sequences and these later sequences. Evolutionary changes driven by the immune pressure by this imposed by this novel antibody. Okay, talked about highlighter. The point is, it takes again sequence alignments and shows just the things that are different. It strips out all the things that are the same. There are a number of more complicated things it can do and are worth thinking about. But broadly speaking, what we use mostly for is, well, there are a number of things we use it for. Recombination is a good test case. This is what it looks like where each of these is one sequence. Everything is relative, in this case, to a consensus. I didn't mean to badmouth them before. It's not good. Not always good tree roots, but they're useful for other purposes. So, given this consensus, you can see, say, well, every tick mark here represents a base that's different from the consensus. Okay, we have 2,800 plus bases, and I think it's bases. It might be amino acids. And you can see that most of the sequences are different here, but the clear thing is that they fall into, there's some stuff going on where there are big clusters here that are different. And if you look at these sequences, and you look at those sequences, those are all consistent with one another, and these guys are a mix. And in fact, what's happened in this set, which is all from one patient, is almost certainly, you see how this guy, this cluster of differences is the same as this, and these guys here are all the same there, and there's a mix of stuff in here. This is a recombinant infection where at least some of these guys came in, and some of these guys, and those are still present in the sample, but there are also these recombinants between them where there's been mixing and matching, which is just one of the ways HIV is hideous. So, so how it's been used in the literature for testing infection multiplicity, which is what I just showed you. So here, if you have a bunch of samples, these are half genomes, or actually this is just all envelope, I think, where these are all sequences from one patient. There's the tree, there are the changes. There are just a few scattered about. This looks like, and can be statistically shown to be indistinguishable from a single sequence infection followed by with a few mutations accumulating. This one, in contrast, where you have two clusters, is two distinct sequences with a few mutations accumulating. And this guy at the bottom is two quite, these are two closely related sequences, whereas these are two quite distinct sequences. You know, this cluster and this cluster, two distinct founders with some substantial progression and recombination between them. So infection multiplicity, a big deal. CTL escape. So this is I think by Jesus, Salazar Gonzalez and others. So this is a time series of HIV infections broken up in half because they did half genomes to 4,000 or 4,500 amplicons, one for each half the genome. In very early infection, this is starting at 16 days post-infection, then 45 days out, and up to 6 months and a year and a quarter out. And what you see here are a few mutations scattered about the genome, okay, followed by consistent stripes here. And it turns out that each of these stripes represents a CTL epitope. So what's happened is sometime between here, 16 days, and here, 45 days, CTL pressure drove complete escape in the population from the originally infected form to this different form, okay, in these locations. Partial escape here. And you can see that developing throughout the course of the infection. And really the frightening thing is how fast the virus walks away from induced immune pressure. And finally, we have, so these are all confirmed CTL epitopes, with nice CTL data. But we also have antibody escape data, similarly. So this is just a nice example of showing how the pathogen evolves in response to immune pressure. Another way of looking at it is sequence entropy, and in this case, this is that same patient. This is a epitope near the 3' end of the genome, and so entropy is a way of quantifying variability. It's based on does anybody ever know what Shannon Entropy is here? Okay, excellent. Well, so you compute Shannon Entropy, which is I'm just going to say it's a function of the variability of any particular sample, and it turns out to be a pretty good proxy for immune pressure. So in each case here we've got control sample, and we've got blue, green, and red for progressive time points at these particular epitopes. And the kind of groovy and frightening, as I say, is that at in this case 16 days post-infection, there's already extremely high variability at this epitope. So in 2 weeks, right, post-infection, there's a good CTL response mounted, and the virus is mutating away from it. At this point, there were something like 45 different variants at this of this dimer, and a few weeks later, that entropy's dropped because it's found in the escape. It's sampled it's sampled a huge number of different variants, different escape variants, and found one that's pretty fit, which has then gone ahead and outcompeted. Okay. I mentioned hypermutation. So here's a highlighter plot, and the thing you might notice about these two sequences on the bottom is there are a lot of green ticks there, right? And those all represent nucleotides that are A here and something else there. And I mentioned hypermutation earlier, as when we were looking at tree tool. So you might say to yourself, that looks like hypermutation. Those are a lot of A's, but it's not just A's that make hypermutation. It's A's in a particular context. And the context looks like this. So we have a tool that will give you a statistical test of whether there's more A's or more G to A mutations occurring in this particular context that's recognized by this particular enzyme. And I'm just saying it's there, and you could use it. Finally, we have these quality control tools so for people who aren't doing it sequence. And it's a nice way of generating GenBank entries. And probably not relevant to this audience. So I don't like to think of myself as a scold, okay, or a moralist, or somebody standing at the pulpit telling you how you ought to live your life. But there are some things that, if you're working with sequences, there are some things you ought to do, and whether they're HIV or not. And some of these things could be generalized to other kinds of data, and probably should be. But I'm going to say, look at your sequences, right? Look at them. They're actually spending hours going blind, staring at your alignment is worthwhile doing because you start to see stuff. And looking at, you have to look at the DNA, you have to look at the amino acid because coding sequences code. That's what they do. And different phenomena happen at different levels. Look at different scales. Remember, you can't trust a machine to do a human's job. Okay, algorithms are great, but you can't implicitly trust them because they will frequently generate absurdities. Build trees include sensible outgroups and other samples from your lab, other samples from other people's labs. Think what could be contaminated in here and find it. Make sure you know how to get back from your figure to your data. Accession numbers and sequence names, always good. Make sure you have enough metadata associated with your sequence names. Submit sequences to GenBank. That's where this is a public resource. Call your mother. She wants to hear from you. Okay? Thanks very much. So how are we talking?
We are. You finished?
Okay, so I just
Okay.
handed out some evals for them, so if you want to take a second to do that, please do so. Otherwise, that's the entire slide deck, right?
That's it.
So perfect. So we have essentially 30 more minutes now to do individual attention. Obviously, since the large slide deck is done, you can stay in here and meet. We also have 1101 and 1103 booked out. I don't know if we wanted to.
Maybe we can stay here and something.
Okay, yeah, so if you have questions, you know who we are, but yeah, it's just kind of hang out or take off with your